Then welcome back after lunch. So I'm your session chair for the next invited talk, 3 papers, and another invited talk afterwards. My name is Ivo Ihrke. I'm a professor for computational sensing at University of Siegen. And now I want to introduce our first speaker, who is Luat Vuong, who is an associate professor of mechanical engineering at UC Riverside. She did her PhD at Cornell University in 2008, postdoc at the Institute of Photonic Sciences in Barcelona. She has received a couple of fellowships, like the Fulbright Fellowship in 2007, NSF Career Award in 2012, 2019 DARPA Young Faculty Award, and the DARPA Director's Fellowship recently. Her topics are at the intersection of optics, photonics, and sensing with machine learning and data science. And today she's going to talk about nature-inspired Full-Stokes polarimetric camera. Thanks. Welcome.
Thank you. It's a pleasure to be here. Can you all hear me? It's on. OK, great. Thank you for the invitation to be here. I don't consider myself a computer vision person, but I'm a fan. And like you, I share an interest in hybrid imaging. For the past 5 years, we've been thinking about this paradigm right here, where we have an encoder on top of a lens, thinking about the compression. And our primary focus has been on the optics. So in my lab, we have kind of a Swiss Army knife effort looking at encoders, compression, and then the decoding. But we only focus on very, very simple machine learning. And we focus more on understanding multi-scale, semi-ordered, varied ordered encoders. And the reason for looking at self-assembled and random encoders is this nature-inspired super resolution, which I'll show in the next slide. So this is a BBC. You can go down a rabbit hole looking at YouTube videos. And they played in slow motion. And then you see the fly go up and come down. You don't know what it actually saw. But then in slow motion, you can see that from a long time, a long distance away, it was able to track an object, this tiny little object, and capture it. And this is so mysterious because if you look at the very tractable anatomy of the fly eye, it only has enough sensors to really sample every degree. Okay, so how could it spot this object from so far away? Well, taking this apart, the fly eye has the fly brain is very small. We might think that a big brain is better than a small brain, but in fact, it's got a very low power brain. If we're talking about low swap cameras for drones, it's a very light brain, has very few neurons, and it achieves very high frame rates. So what else do we know about the fly eye? Probably most of you are aware that the fly eye has a compound architecture, anywhere from 700 to maybe 10,000 lenses, also known as ommatidium. But what is less common is this knowledge of the surface nanostructures that cover, that ornament every lens. And this varies from species to species. And although that for a long time it's been considered that these fulfill mostly an hydrophobic function, so that basically flies don't need windshield wipers, we've started to impute, so biospeculate, that there has some optical function associated with these nanostructures and many other nanostructures in the fly eye. So we have to be careful here because we can't impute too much form and function. These co-evolve together, but they don't necessarily optimize for different environmental conditions. However, we have noticed that there's clustered typologies. That is the physiology that sort of correlates with the behavior of the insects. So, for example, daytime insects have compound eyes where the insect retina is only related to the light coming through a single lens. And then nighttime have a superposition eye. Basically, it's been proposed that this widens the aperture. the neural circuitry and the function of those adaptations vary depending on the light conditions. And for me, one of the most intriguing aspects is also looking at the difference between predatorial horseflies, dragonflies, carrion-seeking, blood-drinking flies, which have kind of random nanostructures on top of often spectral filters, and hovering insects, butterflies, moths, and mostly nocturnal insects, but some diurnal insects, that have these raised nanostructures in these almost perfect but highly granular photonic crystal structures. So the challenge with inverse engineering the optics is that these optical structures are not just on the top of the lens of these compound eye structures, but that they are around the nanostructures all the way down to the retina, and they cover multiple scales. So this is a picture that I've put together. These are different flies. This is a long-legged green fly, and this is a nocturnal fly. And the scale bar here follows orders of 10 magnification. So you have disorder and filtering and potentially encoding on the micron scale all the way to the nanoscale. So how do we mitigate and think about building a camera and trying to solve some of these mysteries? One thing I've done is focused on this very simple paradigm of encoding, sparse sampling, and two-layer, very simple machine learning neural networks. This is almost just optimization. This is a focus on building rapid, real-time, lightweight cameras. And in our lab, we started in 2022 working with these nanostructured encoders composed of PANI, which is polyaniline, not PAMI, but PANI. And these are multi-scale structures that allow us to vary the order and the shape over multiple scales without going into a clean room. And this relationship between order and disorder has, to me, been the most interesting part of our project. I think probably Sylvain Guigan will talk more about this on his plenary talk. The order can be described in many different ways. So I have an ordered structure here. This is self-assembled. What we do is we take a colloid, tiny little spheres. We pack them onto a surface. And then we use this as a template to attach other nanostructures. And by changing the surface surfactant, we can change the relative degree of order. The way that this order is often characterized is with a pairwise correlation. So this is an image processing algorithm where you take two spheres and you look at the separation between all potential spheres and look at the statistics. You can also, as a solid-state physicist, apply the approaches where you look at grain boundaries and the rotation of those grain boundaries. And so we're starting to look at the role of order and disordered in these self-assembled polarimetric encoders. So I haven't mentioned polarization yet, but we're going to start talking about what it means to separate different polarizations, and then how the physical characterization leads to explainable machine learning transitions. And one thing that we've learned recently, the archive should be up maybe tomorrow, is that we achieve polarimetric compressed sensing, compression of the light polarization through long-range disorder of the sample and also some blur. And this is how we think that the fly can have nature-inspired super resolution. Okay. So by Full-Stokes polarization, I'm referring to the ability to separate not just linear polarizations, but circular polarizations. Okay. And not just identify linear and circular polarizations, but you're able to tell the difference in the handedness. So insects don't have the hardware, the physical hardware, to sense circular polarization, but there's an abundance of circular polarization signatures in nature. Photographers know this because they'll often look at birds and they'll change the circular polarization filter, and their skin, their feathers can go from black to red to change the coloring of the vibrance of their color. So this is another version of the phase retrieval problem in the sense that TE and TM, or S and P polarizations, these two linear polarizations that are perpendicular and parallel to the plane of polarization on a surface, are easily separable. However, the relative phase between these two components is hard to measure. Okay? In order to separate plus and minus 45 degrees in right-handed and left-handed circular polarizations, you need something called a spin-orbit interaction. You can't just filter. You can't just use a birefringent material. You can't just reflect or refract. And often it requires more than one measurement if you do use those. So what is a spin-orbit interaction? It's a measurement that it's an interaction that depends on the longitudinal component of the electric field. So you're not in the paraxial approximation. One way to break this degeneracy and separate light, depending on its polarization phase between TE and TM components, is to scatter the light. So with me scattering, right and left-handed circularly polarized light will produce vortices of orthogonal circular polarization handedness. What this means is that the phase of the light that is shifted from the scatter will have different phase on different sides, and this can interfere so that you can find the signatures of the circular polarization handedness. So the challenge with using scattering is that, one, it requires often sub-wavelength structures, and two is that the scattering is often weak and diffuse. Okay. This makes it difficult to capture or to identify the appropriate signatures. So our approach to sensing Full-Stokes polarization has been to use nanostructures produced by the spontaneous nucleation of conducting polymers like PANI. This is what PANI looks like when it nucleates on a flat surface. The structures can be as narrow as 10 nanometers. There's some variation, of course. But the other cool things you can do with it is that you can align it in a magnetic field. PANI is paramagnetic. Secondly, it's electrochromic, meaning if it changes color, as if I put it on top of a hydrochloric acid versus ammonia acid just from the vapor. And for that reason, PANI is also used in electronic noses. Once we nucleate PANI around these spheres, we can get these nanostructures that are conducting and change in refractive index on the order of consistently 50 nanometers. They're often aligned in a certain direction in this nanostructure. And you can see here from this photo that it's mostly clear, meaning that if I looked at the light that is diffracted in a diffraction order, you can see a rainbow, just like a diffraction grating. But you can also use the light that is transmitted at the same time. So you have lots of options for building new architectures in your computational camera. So one of the most startling, exciting aspects of our encoder is that there's spatially bearing birefringence, which is the key to Full-Stokes encoding. So on the top here, I've illustrated what a regular polystyrene solid photonic crystal array looks like when you scatter the light. Light is illuminating is incident, it's vertically polarized, and the light that is diffracted all has the same polarization. This is because it constructively interferes in this diffraction mode, but mostly through refraction. On the bottom, I have my PANI hollow sphere encoder. And here we've removed the polystyrene spheres, so these are just shells. And the scattered light has varying polarization ellipticity around this ring. And that depends on the polarization handedness. So now if I take right-handed circularly polarized light, left-handed circularly polarized light, plus 45 or negative 45, these are kind of the polarization point spread functions in the sense that this is the polarization that will be measured around this ring given the input polarizations. Okay. So to summarize, we basically have a solution-processed, self-assembled metasurface. So a spatial light modulator is not really a metasurface. A filter, a grating is not really a metasurface. What's important about this surface is that there are these non-local kind of wave-guided modes. Okay? The key to this spatial birefringence is that there are these polarization-dependent modes that will then be wave-guided along the thin film of the structure. And this produces a retarded phase. The light is slowed down for that polarization. And then when it exits, you get this mixture and interference that leads to this birefringence, this spatial birefringence. So in summary, we have a Full-Stokes polarimetric encoder. I'll talk now a little bit about how we use this for encoding. So if you just use a linear polarization camera and sweep the diffraction pattern, you'll see that the colors change. The orthogonal plus and minus 45 degree measurements change in magnitude quite significantly. So you can get almost 50% change in those different orthogonal polarizations as a result. You can put different spatial detectors and measure the polarization as well. So their relative intensity from linear polarizers can tell you about the polarization ellipticity. So typically, in order to achieve Full-Stokes polarization, we need 4 measurements. Okay. And most polarization cameras lose resolution in order to achieve that polarization sensitivity. So one question we've always wondered is, if an insect sees polarization, if it's able to sense polarization, it comes at an economic cost. How can it get that cost back? Well, if there's some compressive sensing. Right? If it can somehow encode polarization and pointing or features of the image simultaneously. So we've started looking at the physical features of pointing and polarization modulation. The polarization is encoded in these modulations that occur approximately every 40 degrees if I tilt the sample. And then similarly, if I coat this surface if I vary the angle of the incident light, I can see these interference fringes. They're about every 4 degrees. And this is with a relatively ordered structure. Now what happens when I take a disordered structure? Those fringes disappear. Okay. But we can't forget that those fringes were actually there to begin with. When we run simulations with an experimental data set in this encoder system, those fringes which just aren't there provide the encoding that can be decoded from a neural network. So once we built our neural network, we can start to simulate different optical geometries of the fly eye and define what's a polarimetric compression ratio. That is the error that is from the unencoded pointing error to the error of the encoded pointing error, and the similar relationship of the polarization estimates. And we get something like this. The pointing error has this knee around 20 degrees, which is half the modulation of the polarization modulation angle resolution. If you sample above approximately 20 degrees, the polarization predictions fail. And the best prediction is about half of about 48 degrees. Okay. So basically what this encoder does in this very explainable physical way is that it takes the modulation of the point of the polarization and the pointing, and it changes it to the fringes that can be measured in the diffracted scattered light. Okay. This is a very intuitive approach to thinking about encoding. But the problem is here, we don't get a much improvement in the compressive sensing. Okay. That is, we have 4 measurements typically needed to capture the polarization. And we're only getting, and we're using 2, so we're only getting a factor of 2 improvement. Okay. So what we do now is we add blur. Okay. This picture on the left here is the raw data for right and left-handed circularly polarized light. You can see the noisiness that comes from orderedness because you get these sharp spikes from an ordered material. And then when you make the surface a little bit more random and blur it out, you get these stretched out signals. Okay. The transitions in the neural network presentation. The transitions in the neural network predictions are approximately the same. You get this transition where the failed polarization prediction until about 20 degrees, and best compression at about 5 degrees. But as we blur and as we make the sample more random, we achieve higher degrees of polarimetric compressive sensing. Okay. So there's a lot we can do that we still haven't explored with this polarimetric encoder. Building a camera, one option we have is not just measuring the far field patterns, but measuring an intermediate field where there's a caustic, a speckled caustic. The caustic typically would only change less than 1% if you had a random surface. Okay? But if using these nanostructures that scatter and pool light, we get 10%, 15% changes in the intensity. This means that we can also use the near-field caustics of this polarimetric encoder to build a Full-Stokes polarimetric camera. And we can think about the point spread function as one way to decode the output polarizations, and one reason why we need a minimum of 2 layers in our neural network. All right, close now. So the advantage of self-assembled structures is that we can produce them with surface. It's basically like a paint. We can fabricate them over large areas. So you can imagine entire satellite disks covered with this coating, or coating multiple nanostructures for wavefront sensing. For example, if you have a polarization light source and it has one polarization, and then turbulence from the clouds, which is unpolarized light, we can start to differentiate the polarization signals. You can imagine coating windows for non-line of sight imaging, and maybe even invisible cameras. So we're really excited about the potential for solution process metasurfaces, and in particular, polarization sensing. So to close, we've developed a very much nature-inspired, mostly air, okay, lightweight, mostly air, solution process self-assembled structure that serves as a polarimetric encoder through non-local interactions. Behaves like a metasurface. Randomness and blur are the keys for us, the key parameters for us for achieving higher resolution, nature-inspired super resolution. And the physical characterization enables us to interpret a lot of the encoding and the transitions of the machine learning neural network to ideal Nyquist sampling. I'd like to acknowledge Jiaqi, who is a materials savant who got this working, also Xiaojing and Altai. Thank you very much for your time.
Thank you very much for your talk. So I will pose the first question, maybe while the microphone is going on. So the area where I know that people are very interested in Full-Stokes vectors is super resolution microscopy to image fluorophores and their orientation. So is this something that, I mean, I was struggling a little bit in interpreting your graph because I'm not from your community. Yes. So you had like the separation of the detectors being necessary in some sense for the compression, to achieve the compression that you were looking at. Would it be possible to put them closer to get a high resolution?
Okay, so the question is maybe how would we design this for microscopy applications? Because there's a lot of interest in polarization for microscopy applications. So in microscopy applications, you're typically looking at high numerical aperture light and the near field. And in there, the advantage of using, say, a top-down nanofabricated metasurface is higher because you have very controllable order. You know exactly what you're getting. We haven't considered using these self-assembled structures for microscopy, but I think the reconstruction is going to be less favorable because of the randomness. And I'm not sure what the advantage of compressive sensing is for microscopy, where a lot of times you're analyzing those images, you've got hours. Maybe real time is not so important. So our main focus in application is drone cameras right now.
Okay, thank you.
Okay, yeah. Thank you. Thanks for a really nice talk. I was wondering, what is the behavior of this with wavelength? Are you assuming coherent light in all of this, and what happens if you have incoherent light, or maybe even full visible spectrum or something like that? What happens in those situations?
Okay, so when we've thought about this a lot, because of course insects, if they do any optical processing at all, it would be with ambient light. So sunlight is spatially coherent over microns. 10 microns is what I remember measuring. And with insects, nocturnal insects, the spatial coherence can be on the order of meters. So when thinking about what wavelength and what light source we need, we only really need the surface to be ordered on the order of the spatial coherence of the light. So we can use white light. We can definitely use white light. The diffracted light is spatially separated depending on color. So probably the most immediate application for these grading structures would be some hyperspectral application.
Okay.
We jumped into polarization because it seemed like a harder problem.
Okay. Thank you.
I have another question on scalability of these structures. How easy is it first to manufacture these nanostructures and then also scale them to any arbitrary image size, for example?
The question is how difficult it is to scale them. So we are looking at automating the fabrication process in my lab. It used to be that you had some artisanal hand that would dip and coat the structure. And what we found is that there are ways to use what we will call a draining bin, so that no handiwork is needed. So we're looking to make it easier and make it over larger areas. But currently, and if you look in this community, most of them require that you have a steady hand.
There's one more question here.
In your lab, you manufacture the photonic field. Yeah?
Correct.
And we know that the temperature is a very bad thing in the application range, especially when we're talking semiconductor electronics. And the temperature is a problem. Can you give a comments how stable by the temperature is your result?
Okay. So the question is related to probably fabrication and testing and durability as far as temperature of the film. So we haven't found any huge differences summer, winter with fabrication. But in application, so these films are stable up to 140 degrees Celsius. Okay. I have a hard time convincing my program managers this. Conducting polymers are not known for their performance under extreme conditions, and that's a fair comment. But you can start looking into carbon nanotubes. You can start looking into, so there are other semiconductor materials. You're not limited to PANI. We were really drawn by the fact that you can change the doping and that there's some chemical sensitivity.
So it's not catastrophic problem, the temperature for you?
Yes, that's right. And we've had samples in the lab for 3-4 years now, and there's no problem.
Thanks.
Okay, so then let's thank our speaker again. Thanks very much. continue the program with the paper talks. We're going to have 3 talks each. The first one is learned B-Spline parametrization of lattice focal coding for monocular RGBD imaging. And the speaker is, sorry, I dropped my paper, Jiaqi. The authors are from Tongji University.
Hello, everyone. It's my great pleasure to be here today to present our work. I am Jiaqi, going to be a doctoral student at Tongji University. It is a privilege to stand here today representing the collective efforts and expertise of our colleagues from Tongji University and King Abdullah University of Science and Technology. Today, we are showing method for monocular RGBD imaging, in which RGB images along with corresponding depth maps are captured. It is a computer vision task that has wide applications, including autonomous driving, augmented reality, 3D photography, and object detection, and so on. Compared with capturing RGB images, it is not trivial for the corresponding depth map. Over past decades, a lot of techniques for depth estimation have been developed, including time-of-flight camera, stereo or multiple view camera, and structure light. These techniques are able to measure depth accurately and are well established, but suffer from some drawbacks such as bulky and expensive devices, complex calibration, and high power consumption. Therefore, estimating a depth map from a single RGB image or monocular RGB depth estimation, also known as MDE, attracts more and more interest in recent years. Meanwhile, because the autofocus objects are blurred, the ability to synthesize an only-focus image is desired as well to produce high-quality RGBD image. These 2 subtasks comprise our target tasks, monocular RGBD imaging. Some MDE methods estimate depth exclusively rely on semantic information, namely pictorial cues by deep neural networks. Trained on large-scale datasets, these methods are highly robust and generalizable. However, no physical constraint is modeled. So they lack interpretability and tend to suffer from evident hallucination, as shown in this figure. The second class of methods leverage optical coding. Because the point spread function, or PSF, of an optical system depends on depth. Depth variant PSF is able to encode depth information into blurred images. Then computational decoding, typically a neural network, reconstructs a sharp RGB image and depth map. Furthermore, PSF can be manipulated to facilitate depth estimation. For example, coded aperture makes it more discriminated by adding an opaque pattern on aperture. In lattice focal length, the aperture is divided into several subsquares, each of which is the length patch focused on distinct depths. Another approach is utilizing diffractive optical element, or DOE, to modulate PSF. Apparently, the method can mitigate hallucination by virtue of depth variant PSF. However, optical coding, such as surface profile of DOE, is hard to optimize, which is non-convex and usually involves millions of parameters. Some of them even remain heuristic. Second, the depth variant PSF, which is also spatially variant, is hard to deblur. To solve these problems, we make 3 modifications on existing DOE-based RGBD imaging methods. First, we use the geometry of lattice focal length to initialize the surface profile of the DOE to inherit its capability on both only focus imaging and MDE. Second, we propose to use B-Spline as the parameterization method of DOE to constrain the number of parameters and effectively fit such a complex surface. With these two aspects, the original hard optimization of optical coding becomes easier. Third, in contrast to commonly used UNet, we utilize a transformer-based neural network to perform decoding. Basically, this neural network features global receptive field and can dynamically adapt convolutional weights, making it more competent to deploy depth-variant PSF. Eventually, compared with the state-of-the-art method on this task, we achieved 3 decibel higher peak signal-to-noise ratio for RGB images and 39% less mean absolute error for depth estimation on the testing dataset. Next, I will explain why do they work in detail. In principle, a DOE is placed at the aperture plane of a conventional lens whose surface profile can be designed to manipulate the PSF. Notably, the DOE is initialized with a lattice focal shape. In this way, multiple depths are partially focused, so a relatively high MTF can be maintained across the full depth range, which is beneficial to subsequent deblurring. The resulting PSF also exhibits structural depth variance, which is beneficial to MDE. This figure illustrates the pairwise PSF similarity between different depths. As can be seen, a standard lens suffers from an depth ambiguity in that its PSF varies identically when the object gets closer or farther from the focal depth. But the lattice focal lens avoids this ambiguity by a symmetric focusing structure. The second point is B-Spline parameterization. Basically, it represents an arbitrary surface as a sum of a series of basis functions, and the coefficients of each basis function are optimizable parameters. This formulation of B-Spline is simple and can be easily found. What we want to emphasize here is that each B-Spline basis has a very narrow support, which means each coefficient controls only local shape of the whole surface. This makes it easy to represent complex surfaces such as lattice focal shapes efficiently and convenient to make local adjustment, making the optimization more stable. Here we compare B-Spline against another common parameterization, Zernike polynomial. It is evident in the right figure that B-Spline can fit lattice focal shapes more accurately than Zernike polynomial. You may argue that Zernike polynomial can approximate to any surface given enough terms. In fact, however, due to a numerical ill-conditioned circumstance, the proper Zernike coefficient cannot be solved precisely when too many terms are allowed. More details are covered in our paper. In contrast, B-Spline fits in more accurately when more parameters are allowed. This is due to its local adjustment ability. The following is an illustration of our image processing neural network, namely Restomer. It was a state-of-the-art architecture for image restoration tasks such as deblurring and denoising, while in this work, we have made subtle modifications to perform deblurring and depth estimation simultaneously. As a transformer-based network, it has two advantages compared with common convolutional neural networks or CNNs. First, it can process global pixel dependency, while CNNs have only limited receptive field, making it more easy to utilize pictorial cues for depth estimation. Second, it can perform dynamic convolution, while the convolution weights of CNNs are static. These two properties make it more competent to deal with depth variant and consequently spatially variant PSF and extract depth information. Finally, an illustration describing the entire training pipeline is presented here. We jointly optimize the parameters of DOE and the neural network on an RGBD dataset in an end-to-end manner. In each step, the profile of DOE is constructed with current parameters, and depth variant PSF are computed by a Fresnel diffraction integral. Given a ground truth RGBD image, it is convolved with the PSF in a depth-wise manner, which gives captured or coded image. After the neural network generates an RGBD estimate, loss function is computed and backward propagated to update the parameters. The following is a series of simulated experiments verifying the effectiveness of our method. In this section, final RGBD imaging performance on the testing set with different configurations measured by RGB image PSNR and depth map MAE are compared. Proximity to the lower right corner indicates a better model. The first one is latifocal initialization. It is compared against other 3 initializations here, including zero initialization, random initialization, and a method used in precedent DOE-based depth estimation method, phase cameras 3D by Wu et al. In conclusion, the latifocal initialization we adopted turns out to perform best in terms of both deblurring and depth estimation. The second one is B-Spline parameterization. Totally 5 different parameterizations are compared here, including the baseline, which is a fixed latifocal surface without optimization, and rotationally symmetrical one, and pixel-wise parameterization where each pixel is optimized independently, and Zerny polynomial and B-Spline. Considering there is a performance trade-off between all-in-focus imaging and depth estimation, we trained each model with 4 different weights for MDE relative to all-in-focus imaging. As expected, although having the highest degree of freedom, pixel-wise DOE fails to overtake the others, which reflects the aforementioned optimization difficulty. Moreover, the B-Spline beats all other parameterizations in both metrics, confirming our opinion stated above. The third one is the verification of Restormer neural network. It turns out that Restormer we used, which had around 26 million parameters, obtains a significant performance boost over UNet with around 1.5 million parameters, which is the common size for this kind of models. Restormer beats a UNet with even much more parameters, say 23 million, with an evident margin. The depth map produced by Restormer is more consistent with the captured image due to its capability for long-range pixel dependency and dynamic computation. Finally, we compare our method to 2 state-of-the-art ones, including the fake cameras 3D and the work by Ikoma et al., along with 2 baselines performing MDE on all-in-focus images and RGBD imaging without DOE. The degraded performance of 2 baselines suggests the importance of depth encoding introduced by PSF. Compared to the method of Ikoma et al. And fake cameras 3D, we achieve a substantial performance gain, and thanks to B-Spline parameterization, latifocal initialization, and more advanced network architecture. We also assembled a prototype camera and conducted experiments in real scenes. Our final design has a focal length of 50 millimeters and an f number of 6.3, covering a depth range from 1 meter to 5 meters. The middle figure displays a 3D view of design DOE. It was fabricated through photolithography, and the high map was quantized into 16 discrete levels. Here are a common photograph and a microscopic photograph of the fabricated DOE. We also calibrated the PSF of this camera at a series of depths using a pinhole light source and fine-tuned the neural network using a captured PSF. The captured PSF and the design one are shown in the bottom figure. This slide demonstrates several captured photographs using our prototype camera and corresponding deblurred images and depth maps. It is clear that the neural network succeeds in removing the blurring introduced by DOE substantially and estimates high fidelity depth map in spite of the thin structure, such as the flower stems in the 3rd column and confusing objects, such as the leaves in the 4th column. We also set up a scene to perform quantitative evaluation, as shown in this figure. The depth of each object is computed at the mean of pixel-wise depth inside a manually drawn box. In this way, final MAE and RMSE is 0.129 meter and 0.181 meter respectively, which is a quite satisfactory result. Wait for a minute. To verify the advantage of object coding in terms of hallucination, we capture a scene of a laptop displaying a landscape photograph using our prototype camera and apply a fully pictorial neural network, depth anything, and our end-to-end train model on the captured image. It can be seen that depth anything tends to reconstruct the underlying 3D structure, while our model predicts a relatively constant depth map, which is real geometry of the screen. Admittedly, there are still several limitations to our present work, including downplay of the off-axis aberrations and quantization error of DOE or calibration error. We would like to develop a more physically realistic model involving aberrations and fabrication error in the future work. Besides, we would like to consider the combination with state-of-the-art MD methods and our end-to-end train methods. In summary, our work aims to develop a high-quality monocular RGBD imaging system, which involves two subtasks, all-in focus imaging and MDE. To this end, we propose three modifications to existing methods and solve the problems including initialization and parameterization and dynamic processing of blurred images. Thank you for your attention.
So I think we have time for one quick question while the next speaker is setting up. And so could you switch out already the computers while you're answering the question?
Hi, thank you for the presentation. I have a question regarding data simulation. You mentioned that you convolve the PSFs with the separate depth planes of the ground truth data. But I think one of the limitations of these methods that jointly optimize PSFs with other downstream tasks is that it's very difficult to simulate blurring effects on object edges. So you have occluded regions that contribute to the PSF to the final blurred images and so on. So how did you tackle that issue? Because in order to appropriately simulate blurring artifacts within object edges, you need the full 3D structure of the scene, because occluded objects also contribute to the blurring artifacts at the end. And with just using, let's say, quantized approach with convolving the PSF with each depth plane, you end up with simulations that are, I think, quite far from what you actually measure in real settings. And also, if you want to
could you maybe
so
say what the question
how did
is?
you how did you account for that issue when you tested the network on real data?
I think they can be mitigated by a more accurate calibration and the full consideration of our condensation error of DOE and probably some fabrication error.
Okay, thank you. We can take that offline.
Yes, thank you. I have one announcement to make. The speakers of the last paper, Stereo Knowledge Distillation from DPMV to Dual Pixels for Light Field Video Reconstructions, could the author that is going to present please come to the front row and sit here and prepare for the presentation. Okay, So our next speaker then is Hendrik Sommerfeld from the University of Siegen, who's going to present task-driven sensor layouts, joint optimization of pixel layout and network parameters.
Thank you. Hi, my name is Hendrik Sommerhoff. I'm from the University of Siegen, and today I will present you my work on task-driven sensor layouts. To show, is it fine from this distance? I'm confused. To show you what that even means, I want to start with an introductory or motivational example. Here you can see a typical image from autonomous driving data sets like cityscapes, so street view data sets. The task here is segmentation. So for every pixel, we want to know what's there, if there's a car or a street or pedestrians. And if you really look at these images, most of the time, the area where most of the stuff happens is actually in the center of the image, so towards your horizon line. At the bottom, there's mostly street. At the top, there's sky or vegetation. But the highest frequency of different classes is actually in the center. You also know this as human drivers because hopefully you look more towards the horizon and not at the street or the sky while driving. So the main idea of the paper is for a given low pixel budget to actually put the pixels where they really matter instead of putting them uniformly across the sensor plane. You can see it here. This is one manual layout I created where I put more pixels in the center, both vertically and horizontally. But at least to me, it's not really clear immediately that this is the optimal layout, especially if there's a neural network, so a task network for segmentation, for example, in the picture, because you don't really know what the network needs to work best. So the idea is to learn this end-to-end, this layout end-to-end together with the task-specific network parameters. What do we need for that? We need, first of all, actually sensor layout parameterization, so some way to some parameters and how to translate them to layouts. And then secondly, we need a way to propagate gradients back from a loss function all the way through the network to the layout parameters. Before I go into detail, I want to give you a brief overview of our training pipeline. We start with, we assume that we already have continuous radiance functions for our data set. In our case, we approximate those as high-resolution images because those already exist plenty. Then we have our pixel layout parameters that we somehow, I'm going to explain later, translate to the sensor layout. Using that layout, we simulate the sensor given those high-resolution images and pretend that those the simulated image is actually on a uniform grid. That's why this bird here looks a bit deformed, because in this given layout here, we have more pixels in the center, so the center, of course, looks enlarged if we interpret it as uniform layout. This deformed image we directly give to a task-specific network, in this case, a bird segmentation network. And in order to compare that segmentation to the ground truth, which is also in high resolution, we have to resample it and undeform it again in the end, yes. The first thing we need is a way to parameterize these sensor layouts. It's actually pretty simple in the paper. We define sensor layouts as parameterized functions that map a square to itself. The sensor parameters we call theta in this case. And these functions need to be decently well behaved for our math to work out. Specifically, the most important properties is for every theta, the function needs to be invertible and also differentiable almost everywhere. In the paper, we consider only two very simple layouts. One, this rectangular layout where pixels stay rectangular, and one more complicated one where the pixels can also be curved in the end. And with these properties, we ensure that if we transform a uniform grid cell or uniform grid, we ensure that every cell in the uniform grid has a one-to-one correspondence to the final, to the final pixel layout we have here. That's for the layout parameterization. Now we need to work to actually translate this layout and the radiance functions to colors for every pixel. That's the forward pass. So that's pretty straightforward. You probably know this already. This is a simplified version of the measurement equation. So this is basically just a weighted average of all the light that hits sensor or pixel area. The weighting function W here can, for example, be used to normalize the final color by the pixel area, or can also be used to model a non-uniform sensor response, so different positions inside the pixel respond differently to the light. Yeah. So this AK here is just the uniform cell transformed with our pixel function, layout function. And what's interesting here is that this integration domain now depends on theta. So that needs to be taken into account when we compute the gradients, which we of course need for backpropagation. Yeah, I don't want to go into the math too deeply in this limited time. To actually compute the gradients, we can use the Leibniz integral rule, also known as Reynolds transport theorem. That gives you two integrals to compute the gradient. The first integral looks almost the same as the forward pass, actually. The only thing that's different here is that we now have the gradient of W. And that's pretty easy to compute also during the forward pass. W is easy enough, like in our paper. We can even use the same samples to compute this first integral. The second one is a bit more interesting. It's a boundary integral over the path of the boundary of the pixel. To compute that, we need these vector quantities at the boundary, which are surprisingly also quite easy to compute. More details in the paper for that. Yes, now we have all ingredients we need to backpropagate all the way from the loss function to the pixel layouts. Coming back to the original example from the introduction with cityscapes segmentation, you can see here the layout that was actually learned by our method. As expected, vertically, there are more pixels in the center, but maybe more interestingly, there are also more pixels put on the left and right sides of the image. Our hypothesis here was that's because of the sidewalks. And I think this is interesting because this is not something humans can do. Humans can't focus on two areas at the same time, but here the layout that was learned actually has two focus areas. Here you can see some numbers for the results. In all cases, our learned layouts outperform the uniform baselines. What's also interesting is that the rectangular layout also outperforms the curvilinear ones, which is nice because the rectangular ones are also easier to build. But you can also see that for higher resolutions, the margins decrease a bit. So this it looks like this only really works for low resolutions, which was also kind of our goal. And here you can see the accuracy gain per pixel for all positions on the sensor plane of our rectangular layout compared to the uniform layout. And you can see here there's only few areas, few orange areas where we perform worse, but a lot of dark green ones where the pixels are smaller. And yeah, now I'm also, I'm almost at the end now. Yeah, this paper was more about the optimization process of this whole pipeline. Yeah, I myself know almost nothing about electrical engineering. So luckily, I have some colleagues that actually published a paper on how to actually build the layouts I just showed you earlier this year. You can scan this QR code if you want to check out that paper. Yeah, that's it from my side already. Thank you. Maybe I'll show the QR code a bit longer while I answer questions.
Thank you very much. So I have a question. What happens if your new camera is not mounted properly?
That would be bad. Usually we assume that it's always mounted in the same place. Like in cars, you can assume that, I would guess, because it's always mounted unless it breaks.
But let's say a mechanic needs to exchange the part where it's mounted. Yes, you need to ensure a proper calibration procedure, I suppose.
Yes. Yeah.
Yeah. There's more questions.
My question is that have you visualized the learned layout? I have not seen a visualization of the learned layout. I wonder if it is related to the lens distortion, because usually a lens has some distortion around the edges. What is this related to the lens distortion?
Okay. So sorry, I was distracted a bit.
Yeah, I mean you have learned the pixel layout.
Yeah.
I'm wondering whether it is related to the lens distortion. You have a lens you didn't consider lens.
Light? Lens distortion? No. I assumed pinhole cameras actually. So lens parameters are different, are almost orthogonal to my optimization. You could also include a more realistic forward process with lens simulation, but I didn't consider lens distortion here. Or do you mean I can generate this very layout with lens distortion?
Yes, I think they're highly related, for the large, especially large things.
Yeah.
Thank you.
That might be possible, yes. So if this is possible to emulate with lenses, that would be even better. But I don't know too much about.
Okay, sure.
lenses.
Thank you.
Hello.
Yeah.
So I would have thought the curved representation might do better. Do you have any intuition why the grid one did better?
Yeah, maybe. So I don't really know why, of course, but I think the distortions from this curved layout are a bit too much for the network to handle. Because if you looked at the curved one, we have this circle in the middle where it's actually only distorted, and at the border there are very distorted pixels. So maybe that's a problem for the network to handle. Maybe there are different layouts you can think of that perform better, but we only considered those two in our paper.
Okay, one more question here.
Thank you, very interesting talk. Do you have any intuition on other camera parameters you could optimize end to end? So what's the next step after a layout?
Yeah, we thought a bit about optimizing also color filter arrays, maybe also non-uniform, so different areas have different color filters. So that could be some future work. Because if you look at the sky, you know already there's a sky, you know it's blue, so why put blue pixels there? They don't give any new information, so that would maybe be future work.
Okay, thanks. Let's thank our speaker one more time. And continue with the last talk of this paper session. But then afterwards, don't run away because we will have another invited talk right after. So the current talk is stereo knowledge distillation from DPMV to duopixel for light field video reconstruction. It is given by Raghav Malampalli. The authors are from IIT Madras, Microsoft India, and Simon Fraser University.
Yeah, thanks a lot for the introduction. Can I just confirm that I'm audible to everyone? Yeah, cool. Thanks for the opportunity also to present this work. the work that we're doing primarily is kind of extracting depth information from a kind of sensor called dual pixel, and then performing better light field video reconstruction using that. So this is an attempt to take RGB plus dual pixel data and then directly generate a light field. And to achieve this, we have captured a new video data set, which is DPMV. So first, I'll quickly explain the benefits of capturing a light field. So on the top right, you can see a light field image, or at least an extracted view of it. So the advantage of capturing a light field is that it allows you to do a lot of post-processing, such as generating refocused images or novel perspective views. But it's also extremely expensive. So I think it's been discussed prior. So things like camera arrays or very expensive lens slits between the sensor and the lens need to be employed for light field capture, and the resolution tends to be low as well. So a very kind of, the next step that we can take immediately is to attempt to regenerate a light field from fewer views. So two of the existing solutions from our lab are stereo-based reconstruction, which is self-V, and monocular reconstruction, which we just call mono. There are also some other kind of methods abbreviated into this table, which you can look at a paper to understand more about. So the advantage of stereo is that with the two views, you can set up a baseline and then get depth information, and then follow up with a full light field generation. Whereas we also have the same expense problem of we need a calibrated stereo setup in order to capture the information. So when we go to mono, we eliminate this expense, but then we trade it off for a complete lack of depth information. So we used an estimate, which is a purely semantic depth extraction from an existing monocular depth generation network, and then we pass that into the network in order to generate better light fields. So we were looking for a way, a technique that we've used in this work is tensor display. So this is a physics-based interpretation of light fields, which allows us to apply a strong inductive bias on our modeling process. So you can look at the paper for more details, but yeah, we basically take a low-rank representation and then inject angular and spatial information into it to generate a light field. So what's a dual pixel sensor? So in a regular sensor is displayed on the left and a dual pixel in the middle. So all the green subpixels are sharded into two, and this allows us to capture a disparity in the information between the left and the right halves of each of these sharded dual pixels. So you can see in the right image that there's a change in the defocus deblur depending on where the object is in the scene. And this is kind of what we exploit in order to generate better depth maps. So there are two sensors I've shown, two devices which have this sensor, but there are more coming out as we speak, like Samsung. Their Samsung line also has a lot of dual pixel sensors, and also the Huawei P40. How does this work, right? So we have a lens slit array in front of the sensor, which separates which kind of highlights the disparity between images. So when the object is behind the focal plane, you get more light incident on the left half of the dual pixels, and when it is in front of the focal plane, you get more on the right. So we can use this disparity, intensity disparity, in order to regenerate a depth map, or at least an approximation of it. So this is an approximation of monocular depth. So you can see from this comparison, if you're looking really, really carefully at the right image, that there's a slight disparity. But this is not really very useful for the naked eye, but algorithm can exploit it very effectively to get better depth maps. And that on the left is a stereo baseline. So that's very clearly, the difference is visible even to the naked eye. So the issue with dual pixels is that objects close to the focal plane, as in within the depth of field, don't have these disparity cues, and this is a problem we had to solve in our paper. So we kind of used more semantic information to supplement the physics information that we get from dual pixels in order to solve this problem. So how did we do this distillation, right? We are attempting to distill from stereo knowledge into a dual pixel input. So only the teacher sees the stereo inputs, whereas the actual network, the student, only gets the dual pixels and the RGB data. And then we use priors such as warping in order to make sure that there's a physics-based interpretation for our network's performance. So this is the setup we've used to capture our data. We have 3 different Google Pixel 4s, and this is the image capture extracted from our video capture of 3 parallel views of the same object. So from our data set, we have computed unimatch-based disparity. This is a stereo network in order to generate disparity. You can see that the stereo network is able to effectively extract the depth information for a variety of different objects, and this is one of the reasons that we chose unimatch in order to compute stereo depth. So this is an obligatory cat video. This is Ting with a mess cat. So you can see that at every single instance, we are able to capture and regenerate the depth map, and this is also useful for our work. So first, we look at our qualitative results of the dark distillation hypothesis. So what we see is that the dark knowledge distill networks outperform mono and dual pixel-based disparity estimation, and they also have lower parameter counts, which means they can run on smaller devices. So you can see on the left monocular estimators, which appear to do very well, but then you have the hallucination problem, where objects in the intermediate of the scene are like between the focal plane and the like the furthest infinity are not correctly captured. So you can see the unimatch stereo teacher having branches in the intermediate focal planes as well, which are very well captured by our highest fidelity network, but not by the dual pixel estimators or by the monocular estimators. So this combination of physics and semantics is what is driving the strength of our network. So some more qualitative results for our model. You can see that there is good quality extraction of depth from a purely monocular setup. We also look at benchmarks. So our models handily outperform all the existing dual pixel and monocular depth estimators. We also are able to, our fastest model is able to infer within an impressive 174 milliseconds on CPU. So what is our architecture like? This is a little complicated to look at, but it's very simple in actuality. So we have a Scene-Corpus VID, which is a convolutional vision transformer model. So we use a convolutional model because we want locality information. This is very useful in order to reduce the parameter count and also to remove patch embeddings, which are kind of interfering with the dense reconstruction task. We also use a shallow deconvolutional decoder as like this is a standard. We also have a disparity planes predictor, which attempts to decide how much distance each of the low rank representation light fields are from the actual light box. So the adaptive tensor display layer combines the information from the disparity planes predictor and from the low rank inputs to generate a full light field, which you see at the bottom right. And what are our losses like? So we have 4 different losses: geometric, photometric, bins, and temporal. So our geometric loss essentially warps each of the light field views, which you can see in the bottom right. So they warp that to the center and then check if the difference between the L1 difference between the central view as in the input and the generated light field image are kind of minimal. The bins loss operates on the disparities. And they verify that the stereo computed disparities planes are the same as the generated disparity numbers from the disparity planes predictor. The photometric loss just compares the central view of the output and the input image. And the flow teacher is uses a 2-step loss. So if we first warp the light field image to the center, and then we also warp that 1 frame forward in time using optical flow that we generate from a different optical existing pre-trained optical model. And then we calculate the L1 loss. So this is the overall architecture and how the inputs and the outputs interact. These are the actual equations. I'll just skip over them for the lack of time. You can refer our paper for the actual equations. But yeah, 4 in total. So we see that the it's actually better to not provide the disparity, ground truth disparity as an input to the model, because this causes one of the losses, the geometric loss, to converge before the others, and that reduces the quality of the overall estimation. And we also see that the geometric is the most important loss, followed by the bin chamfer. These are the ablations we performed on the losses that we provide as for the teacher framework. In terms of accuracy, one observation we had was that our work improves performance with respect to spatial resolutions, whereas others plateau because they kind of overfit to the input spatial resolution. So you can see the trend line for ours is constantly moving upwards, whereas the others plateau. And the next is we saw a very impressive zero-shot performance. So we attribute this to a combination of our complex and diverse data set, and also to the strong priors that we have set up using the losses that we use. This is zero shot in the sense that all of the other networks you see, they have been fine-tuned on ground truth light field data, whereas ours has not been done. So we're one of the top 2 performing networks for all of the tasks, even though we've never seen actual light field data. It's purely a self-supervised algorithm. And we also see a very high temporal consistency. And again, we are one of the top 2 networks in most of the data sets that we've tested on. And we also maintain a very low parameter count while doing so. Here we have some EPIs to show the quality of our network. So you can see that the geometry and the tint is preserved in our network, whereas the other networks try to either, for example, in the bottom Lee's work, they tend to add a green tint, which we do not. And above, we see the geometries of the EPIs are preserved correctly. And in terms of some more qualitative results at full resolution, so you can see that, so this is an important thing where in mono, because it's using monocular depth, there's a lot of hallucination and visual artifacts. So we've dropped the intensity in order to highlight these differences, where ours, we don't have these artifacts anymore. There's a lot of JPEG compression, unfortunately, but you can see that our light fields are kind of consistent and they move through the scene correctly. And this is this is matching the ground truth light field baseline. Yeah, sure. So this is, we're trying to scope through, okay, I'll just skip through this for this. And yeah, so to show that our network is correctly predicting the depth planes, we've done some artificial object insertion. And we see that the shadows are correctly predicted in the top right image set. And we're able to correctly predict occlusion information in the bottom right, where Woody is correctly obscured when he's moved across the scene. And in the bottom left, we are able to repaint, recolor, and also insert objects as well. So these are just all of our novel contributions. Thanks a lot. That's the QR code to the paper. Any questions, please?
Thank you. So I ask a first question. What happens if you actually close down the aperture of your dual pixel camera? Do you see vignetting in the images appearing?
We've never tried this, but yeah, I would assume there would be some amount of vignetting. But it behaves very similar to an RGB camera in the sense that it's just half of the camera. So we can model it pretty easily.
It would probably be shifting, I would assume. But,
I would have to look at the physics, but yeah, I assume there would be some of it. Yeah.
Okay. Are there more questions up there?
Hey, it looked like you don't have a light field for the blue and the red channels. Are you ignoring the light field, or does your lenslet array not distribute light across those channels?
So this is an existing sensor, and I think they've observed that sharding the green subpixels is what provides the best performance. So yeah, they only look at the green one. So that's why the dual pixel sensor, the images that come out also look grayscale, because they're only looking at the green intensity.
Gotcha, thanks.
Is that it? Yeah. Thank you to everyone who helped generate the paper. And so a special thanks to Doctors Viragavan, Sustruk, and Ashdu, because they helped us get an emergency visa for this. So yeah, thanks a lot. Yeah.
Okay, so then we're getting ready for our next invited speaker, Professor Emma Alexander. She could not be here with us today in person because of these computer problems that you may have heard about, so the flight got cancelled. And she joins us online here, luckily. It's early in the morning. So I will introduce her to you. She is an assistant professor of computer science and electrical and computer engineering at Northwestern University. And she's been before a postdoc at UC Berkeley, did her PhD in computer science at Harvard. Exactly, and has also an undergraduate background in physics and computer science. So she's won a number of awards For example, the 2016 Best Student Paper Award at ECCV, the Best Demo Award here at ICCP in 2018. She received a CIVO Fellowship from the Center of Innovation and in Vision and Optics at UC Berkeley, and she's a Fulbright specialist since 2023. Her topics are bio-inspired and physics-based computer vision, and she's going to speak about reverse-engineered evolved cameras. Welcome, Emma. Thank you.
We need to set up the loudspeakers first. Just a sec. Can you try talking again? Okay. And now again? Okay. Again, sorry. Can you try? Oh, no. Can you try again? Oh, my God.
Maybe we try to put the computer here and then the microphone.
We could do that, yeah.
I mean, it's not optimal, but...
No, I don't think that's the case.
Let's try if this is reasonably good. Yeah.
Testing, testing.
Okay.
Can you hear me now?
Yes. Okay, can you talk? Okay. Again? Wow, my God.
I can now switch to that system, I guess.
And now?
Hello, hello. Can you hear me now?
Yes.
Testing, testing.
Yes, we can.
Hello, hello.
We can.
Hello. Okay, thank you so much. Is this good?
Yes.
Awesome. Okay. Well, thank you all so much for being here, and thanks especially to Raphael for managing all of the logistics that got thrown at him last second, in addition to all of the amazing support he's been giving us all along. So I am calling in, unfortunately, from just north of Chicago. I'm at Northwestern, where I run the Bio-Inspired Vision Lab. And I just want to tell you guys a little bit today about reverse engineering evolved cameras. I think it's a really powerful paradigm that could help us a lot. So just to take a kind of a step back, right, what makes what we do special is that when we let light move through a scene into a detection pipeline, so that's optics and sensors, through some computation to give us scene information, we get to have a lot more fun than other people who do this because we tie the detection and computation design together in service of specific scene information that we care about, right? That's computational imaging as opposed to things like mainstream computer vision. So this can look a couple of ways. Sometimes what this looks like is beefing up the computation to exceed the limits of the detectors. And sometimes it looks like moving some of the complexity from the computation into the detection. These are smaller boxes because this is particularly true for constrained applications. This is the kind of thing we're going to be looking at today. And I would argue that as like fun and exciting and vibrant and cutting edge as this approach to visual sensing is, it's also just the normal boring way for vision to work. This is because all biologically evolved systems are computational imaging systems, right? Eyes and cameras do not and cannot evolve independently. And so every successful natural vision system is an example of a computational imaging system. This is great news for us because it means that there are tons of proven successful computational imagers that are out there in the world for us to take advantage of. And these have a lot of different kinds of advantages, right? It's not just benchmark success or accuracy on every single pixel. We're looking at a landscape where there's a lot of design diversity, right? So 500 million years roughly of visual evolution have identified a lot of local optima in this space where we have a lot of cool eye designs that go along with computation, much of which is not yet understood, but they indicate places where promise exists. Additionally, all of these systems have many really nice characteristics that we oftentimes don't build into our technological imaging systems. So, for example, they're all spatially compact. They're all running on really small power budgets because acquiring calories is a risky venture. They're all running in real time because detecting a predator too late means not detecting them at all. And they're really adaptive. They're really robust. So they can do things like one-shot learning, handling domain shifts. We don't see things like adversarial noise or like this kind of brittleness in purely data-driven systems. And so these are all things that it would be really nice to bring into the next generation of computational imaging sensors. I'll point out that a lot of these metrics for success are not like pixel level. They're not get every single thing exactly perfect. They're not compete for a 1% increase in performance. They're really different axes of success. And so I think the bioinspiration piece not only gives us a bunch of really promising avenues, but it reminds us to be thoughtful about what we consider success. We should measure what we need to measure with the constraints that we have. And this gives us a lot of different ways to think about that. So the way that I think we can bring all of these advantages into our devices is by understanding vision holistically. And that means not just doing forward engineering, where we take principles from computing and physics into devices, but recognizing that there are successful devices out there already that we can reverse engineer with the tools of vision science. And I think if we can sort of work this forward and reverse engineering cycle, it's this really sort of virtuous cycle where each half can build on itself, and they can really boost each other and move each other forward. So some systems that I'm particularly interested in, I worked on defocus in the jumping spider. That's what we're going to talk about today. Also, if you don't like spiders, maybe take a coffee break. We're going to see a lot of them. I've also looked at motion in the zebrafish, stereo disparity in different parts of the monkey brain, and I have a number of projects going right now on color perception in a number of different animals. So today, we're going to be sort of taking as a case study what we've managed to learn from the jumping spider. So here she is. This is a jumping spider. She's called that because she jumps. And they do that to get prey, to get from place to place, to escape predators. And here's a funny behavioral test that came out a few years back now. So under green light, which the spider's expecting, you can see they jump and they catch their food. Under the red light, this fly's a little blurrier, but here it is right here. They jump short, right? You have to do a little sort of second jump to get to the fly. So we'll just see that again. We get the prey under green light. Under red light, we jump short. And it turns out that the amount that the spider jumps short is exactly what you would predict from the chromatic aberration of the lens. So behind these these main eyes. They have this stack of retinas. And the sort of surprising thing that they were able to conclude, right, is even though they have all of these different eyes and they could be doing stereo, for example, with these principal eyes, they're fooled by this chromatic aberration. And the biologists use that as a strong indicator that the spider is using defocus. And they said there's this thing called depth from defocus in computational imaging, computer vision, and they said mystery solved. And we looked at this and we said, wow, mystery's not solved at all. This animal has a really tiny brain, and we usually think of depth from defocus as being really expensive, certainly more expensive than stereo, which again, they could be doing with their eyes. They move independently. So when we look at this, we were pretty confused because this doesn't look like a successful depth from defocus system in a number of ways. So first of all, maybe most importantly, it's got a brain the size of a poppy seed, right? So about 100,000 neurons total, 1 cubic millimeter. This is like one hypercolumn in V1, if you're familiar with that part of the human visual system. Additionally, their eyes don't look right for this either. So a lot of times our defocus-based systems will have coded apertures put in there. We can look at their eye and see that that's simply not the case. They have a regular open pupil. And then the funny thing is that this defocus change is actually not, it's caused by a funny little mechanism, or it's measured unusually. So in the back of this eye, they actually have a stack of 4 translucent retinas right on top of each other. Because they're right on top of each other, it means they're experiencing a small change in defocus. Now, typically you want the source of your signal to be large. And so usually depth from defocus, you want a large change in defocus to give you a strong depth signal. We said they can't be getting that. They're right next to each other. And that turned out to be sort of the thing that unlocked all of this. Okay, so we're in this situation where we have our mystery, our motive, and our clue. So our mystery is, how is this doing depth from defocus if it doesn't look like a depth from defocus system? Our motive is, if we can solve this, we'll have a cheaper way to get depth out from defocus than anybody's found before. And our clue turned out to be this very small change in focus. So it turns out if you consider a differential change in defocus, there's a whole class of really efficient depth sensing algorithms that get unlocked by this, this idea. So here's an example of what this would look like for the spider eye. Our imaging model is really simple. We have a thin lens with Gaussian blur. So if I give you a scene, I can write down directly what that image is going to look like. And if I just move the sensor back and forth, I'm going to have a slightly different image. So 2 changes are happening here. The first is that there's a little bit of an expansion, right? Because I'm now a little bit closer, a magnification change. And the second is that the amount of blur will change as I come into or out of focus. So it turns out that I can express both of these changes in terms of image brightness derivatives. So I'm going to call IT this per pixel change. I'm going to think of it as a time derivative because our cameras, we'd have to move back and forth. For the spider, they're measuring both of these instantaneously, but this is just retina 1 minus retina 2 is our per pixel change in brightness. We can also capture the magnification by taking this radial derivative. So just the X and Y derivatives put together in a weighted ratio, or a weighted sum based on where you are in the image. And then the last thing I'm going to need to measure is the defocus term with the image Laplacian, so the sum of the second image derivatives. It turns out once I measure these 3 things, the depth at that pixel is just going to be a weighted ratio of those 3 things. So here's my algorithm. At every pixel, I apply a small number of filters. I throw them into this algorithm, and that gives me the depth at the back projection of that pixel. I do this in parallel everywhere, and I can get a depth map of the scene very efficiently. There's a bunch of different ways that we can do that. So that's what it looks like for the sensor location, like for the spider. We can also consider squinting the aperture, changing the optical power. That's what the human eye does. We can move the lens location, like in a lot of cell phone cameras. You can take a regular camera and just move it around or mount it on a moving robot or have it totally static but observing a moving scene. So just motion alone can do this. And then you can start to combine things, like maybe your hand is moving a little bit while your phone is focusing. You know, any small change in defocus, basically, if we can model it, we can get the depth out. And in fact, if there's velocity as well, we'll recover the velocity simultaneously. All of these end up looking a little bit different, but they follow the same basic form. You have a pair of images that are differentially defocused. You apply a small number of filters to get derivatives. You plug that into an explicit model, you get out a depth map, a 3D velocity map if that applies, and then also confidence, which I'll go into a little bit. These all look the same because they're all following the same underlying constraint. You can think of this as brightness constancy, but with a defocus term and with the terms kind of broken up a little bit differently. So what this says is if I measure my image change and my spatial derivatives, so this is all stuff I can measure directly from the image, these are going to be weighted relative to each other, and those weights are going to reveal the information that I care about. So we get translational motion out of the first two weights, magnification change, and then the defocus change. So for the spider that we saw, there's no translational motion, the magnification is hard-coded, and the defocus change gives you the depth immediately. So our algorithm is just this per pixel equation. On sort of the other end of the scale, we have a standard camera that's moving, and then you get the depth magnified motion vector with a defocus weight that sort of breaks the ambiguity. So you solve this small linear system over a patch, and then your depth and 3D velocity are one-to-one with the weights you recover. This gives you really fast algorithms because of the form of this equation, right? I'm only considering local filters, and I'm requiring a linear system. It turns out that if you require that form of an equation, the only analytical solution is for a Gaussian filter. So that starts to explain why we don't see an aperture code helping these animals out. Okay, so here's an example of what this can look like. We take accommodation, it's the change in optical power. That's going to be similar to the spider. It's going to be a per pixel depth equation. This is also how our eyes focus. And of course, you can buy a deformable lens that can accommodate under electrical power. So we built a prototype here that fluctuates your optical power, changes where you're focused in the world, and we sync the camera capture to the peaks and troughs of that oscillation. And this is able to give us depth and confidence at 100 frames per second on a laptop. So just a quick look at what this looks like. Up at the top, we have a top view. Down on the bottom, on the left, we have what the camera sees. On the right, we have the output. And the confidence here, you can see there's a lot of black pixels. We're not doing any kind of fancy image segmentation. It's just that because defocus is a triangulation cue, you need image texture. That texture can even be stuff like reflections off of a transparent object. Here we have a deformable object, but one that would be difficult to measure with a lot of different depth sensing algorithms, but for us, all of these reflections are just more texture. And then anywhere that we see that we don't really have spatiotemporal derivatives or a lot of contrast, we can get an answer, but we know it's going to be garbage, so we clip it out. The advantage to this is that, as you'll see here, we can see multiple depth planes simultaneously. We're not trying to sort of falsely interpolate or guess at pixels where we know we have bad information. So this is an exciting step forward, but if we look at this camera, it's kind of a lot. So there's this camera, there's the control electronics, there's this big laptop. When we compare it to the jumping spider, we see that we have a long way to go on things like overall power efficiency or like the compactness of the device. So we're starting to make some progress in this direction. One of the things that we did was to take away all of that optical complexity and sort of control electronics on the camera by using a metal lens. So these are nanophotonic devices made up of tiny little pillars. You can change their shape, orientation, size, and essentially put in a phase delay pattern that you can design. What's cool about these especially is that they're smaller than the wavelength of the light. And so you can actually multiplex different optics into the same metal lens. So we do that. We built a sort of an interleaved pair of lenses in the same optical setup. And what it does is it sends light with one focal plane to one half of the sensor and with another focal plane to the other half of the sensor. So we're simultaneously capturing multiple defocus levels, just like the spider would, except now the complexity is in the optic instead of on the sensor. So sure enough, when we make this, we can do, again, our fast depth and confidence measurements, even for difficult objects like moving water, which again would be hard to measure with a lot of other depth sensing mechanisms. Okay, so we're still at 100 frames per second, but now we're 30 times lighter and we have a passive optic. We're also, trying to bring down the electronics budget. That's sort of our current frontier on this. So just to take a step back, I said, hey, this forward and reverse engineering cycle can be really valuable. We started with a mystery in animal behavior. We were able to extract a depth sensing principle that was new and exciting. We were able to use that to get depth maps, both with a normal standard camera, with an accommodating lens, with a new meta surface. And again, our current work is to bring this into like a true small footprint, low power, like custom electronic whole package sensor. So that's great on the application side. But it's important that we also are contributing back to basic science. So I'm going to talk a little bit about an optical connection that we made, and then also what this means for our understanding of the spider's biology and behavior. So differential defocus answers the question, what happens to your image intensity when your object depth changes a little? So we came up with this answer. We said, yeah, the intensity derivative with respect to depth gives you out depth by comparing to the image Laplacian and the spatial derivatives. At first glance, this looks a lot like the transportive intensity equation from phase microscopy. So phase is sort of like thickness, which is sort of like depth, and these sort of look the same because they have these spatial derivatives, and they're both answering the same question of how intensity changes when your object depth changes a little. Unfortunately, they're really not saying the same thing at all. So just sort of as a first pass, if the phase, this phi term, is analogous to depth, then we've got our derivatives in the wrong place, right? So on the left, we just take image derivatives, divide them out, and get depth directly. On the right, you've got this second and first derivative of the thing. You have to do this integration with boundary conditions. Like, it's not actually talking about the same thing at all. But they are still like both dealing with the physics of light. So they should be able to be brought into agreement. And we do this with the Wigner distribution function. I'm not going to be able to go into the details, but to give you sort of a high level picture of what we're talking about, and I should mention this is extended not just to fully coherent phase microscopy, but to partially coherent phase microscopy. So there's like a generalized notion of phase that we're going to be using. So let's think about what happens in cameras and in microscopes for defocus. So camera defocus, objects are going to change size, and you're going to lose high spatial frequency content, just like we saw before. In phase microscopy, as you go out of focus, not only do you not have that magnification change, but you can also create new high frequency content in the image. So very different things are happening here. To put them in agreement, we have to make some assumptions to pull them together. So we consider a differential change in defocus under telecentric projection and partially coherent illumination. And it turns out that the transportive intensity is a general equation, and the depth from differential defocus, our work, is a special case. To reduce them to each other, we have to make a few assumptions. The first is that you have point source-like objects, so that this is like a limit on how coherent your light can be. We're also going to assume Gaussian blur. And then this gives us a new interpretation of generalized phase that relates to depth, but has some other things going on in there as well. I'm not going to go into the details, but if you want, it was one of the years that ICCP was virtual, so you can find that on YouTube. And if you're not already subscribed to the ICCP YouTube channel, please do that. Okay, so we've tied this back from spiders jumping around to let's make some new cameras to here's something in a totally different realm of optics that we can connect to and understand more deeply. I think another thing that's really exciting about this is that we can finally explain spider behavior, right? Like why would the color of the light mess them up so bad? This doesn't make sense in the world where depth from defocus is more expensive than stereo. So to sort of explain this, we're going to consider the compute in terms of floating point operations per pixel for similar performance of multiple algorithms. So similar performance, you saw our depth maps were pretty sparse, they were pretty noisy. We think that's enough to enable a lot of behaviors. It's not necessarily what these algorithms were originally optimized for. So we're going to turn down some of the bells and whistles on a recent depth from defocus algorithm that we think has potential to be really efficient, compare it to an efficient stereo mechanism. And when we look at these numbers, we go from about 8,000 to 7,000 flops per pixel. So this is not maybe as big a gap as you might expect. Perhaps some focused engineering could bring it even smaller. But compared to our method, we are 10 times cheaper than previously developed efficient stereo mechanisms. So finally, we live in a world where it makes sense that the color would mess up this spider, right? It's got a very small brain. It's going to really need an efficient computation. And we found the only spider plausible algorithm. Just to sneak preview a little something here, we have one coming soon that's another 10 times cheaper, and that's going to be coming out of the Guo lab at Purdue. So Shi Guo worked very closely with me on all of the cameras I've shown you so far. He's a great researcher, a great guy. Keep an eye out for Chi, and hopefully next year's ICCP we'll be sharing some more details on this new method. So here we are. We have a possible explanation for what the spider's doing. We have the first spider plausible algorithm. Do we know for sure that this is what the spider's doing? We don't. So we are pursuing, with a group of spider biologists, a few more answers to this question. So the sort of key question here is depth from defocus, typically expensive. Depth from focus, typically very cheap. Previously, spider biologists were pretty confident that they weren't able to move that whole eye tube back and forth. So it really was depth from defocus and not depth from focus that they were doing. But now folks are questioning that. So we went down to Argonne National Lab right outside of Chicago with a bucket of spiders. They've got a particle accelerator that throws off really high energy X-rays. And they allow us to, for the first time ever, look inside of the spider's head and see what their eyes are doing as they respond to stimulus. So we showed them a bunch of little videos, their eyes moved around. We're still processing this data, but if you want to check out a little bit more of the rest of the team, the biology side and the motivation for this, that is also on YouTube now. Okay, so we had a behavioral mystery. We got some good math out of it. We got some good applications. We connected back to the basic science. Here's the thing. I don't think that this is unique at all to the jumping spider. I think you could do this for many other visual behaviors within this animal and across a number of different animals. Again, for some pretty big prizes listed over here on the right. So I think especially as we're moving forward into pushing beyond the optical and computational limits that we think our systems have, pushing into really highly constrained things like edge computing, sustainable vision, we're going to need to start taking these approaches more seriously. And I think there's two sort of big ways that we can do this. The first is to look out for gaps between what we think an animal should do and what it is doing. So if it's either really much better or much worse than you would expect from your knowledge as a computational imager, I do think that our field is the field that has the tools to notice these gaps. The thing that sits between anatomy, whether that's optics or brain size or whatever, and behavior is computation. So anytime an animal's behavior doesn't make sense, there's something about computation that we can go and discover. I would say the other thing to look out for is matching the ecological niche. So again, computational imaging, it's happening all around us all the time in a bunch of different settings for a bunch of different reasons. It's been doing that for about 500 million years. And I think that as we're moving into really constrained devices with specific behavioral needs, it's worth just looking around to see what already exists in the world that meets those constraints and had to do this behavior already. So with that, I'd love to thank everybody who helped me with this, and I'm happy to take questions. Thanks so much.
Okay, so this is actually going to be a bit complicated now, because the microphone that we use to communicate with her is here in front of the in the laptop. So people that would like to ask questions, I would ask them to come to the stage and basically speak into the microphone here to ask their question. And then we would be hoping to hear the answer through this microphone into the room. Yes.
Thank you, Raphael. Sorry, everyone, for the drama.
So please come ahead. No questions.
Maybe the stages.
Okay, their Kuros is coming. Yes, you can see us. Okay, so go ahead.
Hey, Emma, great talk. The spiders have lots of eyes, so what about the rest of them?
Good to see you, Kuros, even though you're tiny. And I should point out the depth from defocus algorithm that we compared to was out of Kuros's group. Yeah, so they have 4 to 6 other eyes that seem to mostly act as like a peripheral vision system. So if something interesting happens, they'll turn their bodies around to look at them with the main eyes. So I think anything anything sort of peripheral versus phobia, that's probably what's happening there. And then even within the main eyes, there's 2 UV sensitive retinas that we haven't even thought about. So there's tons more to learn about the spider.
I guess a slightly follow-up question. So what about the neural system that is connected to the eyes themselves? I mean, is there any indication of how these potential computational methods are actually implemented biologically?
That's a really great question. So the spider, unfortunately, there's not many neuroscience tools to go and confirm. That would be the best thing to do, right? Unfortunately, there's not many genetic tools, and they're also, their jumps are hydraulic. So they're actually powered by like muscles in the abdomen that kick out the legs. Really cool mechanism, but it does mean that if you pierce that exoskeleton to measure directly from neurons, they do not last very long. So this is unfortunately not an animal well served by current neuroscience tools. But yes, I would absolutely love to sort of find a circuit in the brain and see what they're doing.
Thank you.
Thank you.
Hey, Emam, wonderful talk. It's Sabina. I was intrigued by this chromatic aberration experience totally in the beginning. So what happens then with white light, right? I mean, it's relatively unusual to be in green, or do they miss a little bit, or what? Or is the resolution down, or what happens in white light?
Yeah, thank you, Sabina. Great question. So these animals didn't evolve to jump under laser light, which is why it's not a problem for them to get the red jump wrong. That retina is mostly sensitive to green light, and sunlight is mostly green. So presumably there is some error in that jump that's probably just compensated with by like the length of their arms. But yes, if they had to live in an environment that was just lit by red laser light, they wouldn't last very long. And this is thought to maybe explain, there are some very, very small animals with red and blue stripes. And this was previously not understood, and now people are starting to think that it's a depth camouflage to fool jumping spiders specifically.
Oh, that's interesting. Thank you.
Thank you.
Hi, Emma, it's Luat. How are you? Great talk. Question. I love what you're doing with trying to find the biological proof, because that's a lot of pushback we get from entomologists or people who actually study the biology very carefully. Tell me how you came up with this idea for the X-ray and how the X-rays actually interact with, like, you can see how the insect, like where the insect is gazing, is that right?
Yeah, so the thing that we were looking for was, so if you sort of look at them through the side, so we were just like shooting them through the temple with these X-rays, and what we were looking for was if that eye tube moves back and forth, because if you're guaranteed to see one in focus image, then all of a sudden we don't need all of this new math. You can just say, yeah, like take some contrast and find a local contrast maximum. That's been known to be cheap for a long time. And so previously, yeah, all of the biology papers just said, hey, they don't do this. But as it sounds like you're quite familiar with, things can change. And so we were looking to examine that. So from their side, they were looking at it in terms of the behavior. We were just kind of interested to see, like, you know, did we do all of this and the whole time it was just depth from focus? We don't think that's what's happening, but we did want to just verify that.
And is the work on the X-ray published yet?
Not yet. No, we're still processing the data. It's pretty gnarly data, to be honest.
I imagine
but I'll
really
send it to you once we have it done.
Absolutely. Thanks.
Thank you.
It was a very nice talk, and I enjoy how you casually refer to walking to a national lab with a bucket full of spiders. But my question is, when people do human depth studies, you come up with these images that are sort of in conflict with respect to depth and stereo cues or something like this. And I suppose it's very hard to get a spider to look at a monitor. But is there any line of research where you construct these sorts of impossible images and try to see what it's doing, or do you have to?
Oh, that's a. Sorry, yeah, that's a great question. Yeah, so humans, we have a luxurious amount of brain. And so basically every person has a weighted sum of different depth cues that they're using, and it varies from person to person, and I think tends to be kind of constant over time. For the spiders, they've got this tiny, tiny little brain. So we think it's possible that they are also computing stereo. I think the color experiment at the beginning shows that even if they are using stereo, the defocus dominates, at least enough to fool them under that condition. We did try, so their retinas are reflective, so you can do eye tracking. And I had this grand plan in grad school to like do eye tracking and like present just the right amount of blur at just the right place and get them to jump the wrong depth. And it turns out that biology is really hard and biologists should do it. And we just had like a closet full of spiders that turned into lab mascots. So to answer your question, I don't have the experimental skill level to do that kind of experiment. But I do think the like free moving behavior is a clue that defocus is at least more important for this one species than stereo. I don't think we can totally rule out that they might also be doing stereo, or that across the 9,000 different species of jumping spiders, somebody's doing something different. Thank you.
Okay, thanks a lot. So I think that sort of concludes the question session, and we would have a poster fast forward session coming up right now, which is going to last 10 minutes. So still 10 minutes of patience before the coffee break, and we're setting up right now. Thank you again, Emma, for this.
Thanks so much. Have so much fun.
Let's hope for the best. All right, is this working? Yes. So we'll hope for the best for this audio with a different laptop. But so I'm Eric, I'm doing a PhD here at EPFL, and with Christina, sitting over there, and Aditya, we're the poster and demo chairs for this year's ICCP. So what we have for today is the first session of posters and demos. So if you haven't checked out the websites and haven't figured out where it is, it's essentially this drop down here. So you'll see there all the posters for day 1, day 2, and in red are the spotlight ones, which we'll be looking at those 1-minute videos today. And the posters will take place on the 2nd floor. So if you go out this way, the stairs should be up that way to go upstairs. There'll be people in the blue shirts to help you. So let's switch over to, let me just check my audio settings. Maybe that will make a difference. Okay, that's. And let's hope for the best. Yeah, I guess I have to do the same thing. Strange. And so all these are on the YouTube channel that Emma also mentioned.
Introduce Doppler time-of-flight rendering. Okay. We introduced Doppler time-of-flight rendering, an extension of time-of-flight rendering for dynamic scenes with applications in simulating Doppler time-of-flight cameras, which is used for velocity estimation. There have been various time-of-flight rendering works, but they are not adequate for Doppler time-of-flight rendering since they did not use dynamic scene or super high-frequency modulations. To simulate Doppler time-of-flight cameras, we derive physically based path integral expressions on the global illumination and compute these integrals efficiently using antistatic sampling and path correlation. Overall, our proposed method outperforms the others in maximum 2 orders of magnitude. Here are more animated results.
Departing from conventional computer vision systems, which first focus an image onto a sensor and then run neural network inference on the sensor readout electronically, we were wondering if we could move this computation to the optical side in an energy-efficient way. To bridge this gap, we inversely design metasurfaces consisting of millions of nanopillars to focus light and perform feature extraction optically. By spatially multiplexing feature maps with a metal lens array camera, we realize AlexNet level classification performance with only 1% electronic compute. This is achieved by exploiting the large kernel spatially varying convolution that can be implemented purely in optics. We prototype the optical neural network as a metasurface array camera that works under incoherent ambient lighting conditions, in contrast to the common diffractive optical networks that only work under coherent illumination. It shows the first time an optoelectronic neural network can compete with the first modern deep neural network, AlexNet, with only 2K digital parameters.
My topic is direct time-of-flight image sensing based on indirect time-of-flight image sensors. Charge modulators are key device of indirect time-of-flight image sensors that can perform inner product operation between the instant light waveform and the applied exposure code without any in-pixel processing circuit. We have developed many kinds of charge modulators with many types. Conventional LiDAR image sensors have pros and cons: low pixel count or vulnerable to multi-path interference. We have proposed pseudo direct time-flight image sensing based on indirect time-flight image sensors using compressive sensing in the charge domain. This is our prototype sensor. This is a demonstration of capturing dynamic scene with 2 bounces.
In this work, we present the method of efficient depth imaging with temporal compressive sensing. Time-of-flight imaging method is a 3D measurement technique that estimates the depth by emitting light into a scene and measuring the time at which reflected light reaches the camera. The direct time-of-flight method is difficult to achieve high pixel counts due to the complexity of the circuit structure, and the indirect time-of-flight method has large errors due to the effects of multiple reflections of light. Therefore, we estimate the depth by observing the temporal scene response with temporal compressive sensing using a coded shutter sensor. This method uses a temporal coded shutter sensor based on a conventional indirect time-of-flight sensor to capture the scene response with temporal compressive sensing and estimate the scene depth by the neural network. This method is less affected by multiple reflections because it observes the time response of the scene like the direct time-of-flight method, and it has a lower observation cost because it uses the indirect time-of-flight method-based sensors.
Hi, I'm Guangyuan Zhao. I'm glad to introduce our work of neural lithography. This is a joint work with Cheng and Peter So from MIT. Lithography systems from various manufacturers have applications in printing semiconductor chips and optical devices. How to digitalize a real-world lithography system? The conventional method models lithography system in a white box manner, but it lacks adaptivity and fidelity. So our first contribution is to use real-to-sim gray box modeling of photolithography system. It will incorporate process data paths. Our second contribution is to close the design to manufacturing gap of computing optics by co-optimizing the design and manufacturability. We show that our method achieves improved results in the downstream tasks such as computational holography and computational imaging. Please come to our poster and know more. Thank you.
In this poster, we discuss the limitations of hyperspectral imaging from RGB images from a data perspective. The effectiveness of emerging data-driven spectral reconstruction is disputed. A simple example here shows such methods struggle with objects of the same color but different spectra. In this work, we design extensive experiments to study the limitations. First, we test the existing methods with slight variations of the data and show these datasets lack diversity in nuance parameters and scene content, leading to atypical overfitting issues. Second, we introduce metamerism and show existing methods all struggle from metameric failure, which has gone unnoticed so far. Third, we investigate the possibility of utilizing chromatic aberrations to partially mitigate the limitations. Welcome to our poster for more details.
This algorithm can see around corners by extracting information stored in shadows. Here, the observer is not able to see the room directly, but by using a digital photograph of the ceiling, our algorithm can reveal the contents of the room. Here's how it works. Each object in the room casts a unique shadow. The shape of the shadow depends on the object's position, shape, and color. We then solve an optimization problem to estimate the three-dimensional scene. Here are some experiments we performed on real scenes where we captured their corresponding ceiling photograph and ran our inversion algorithm. Using only the images shown in the middle column of the shadow cast by the doorway onto the ceiling, we fully reconstruct the scene in 3 dimensions. Thank you.
We introduced RePro LFM, a reprogrammable Fourier light field microscope. It concerns the quest to record whole brain neural activity of lava zebrafish as a pivotal vertebrate model. It is challenging to balance the trade-off between speed, volumetric coverage, and resolution in volumetric imaging techniques. Our RePro LFM addresses this issue by integrating a spatial light modulator and liquid lens to conventional Fourier light field microscopes. It successfully captured 3D PSF stacks and light fields of complex biological structures. And it enables 3D reconstructions via deconvolution techniques. Please stop by our poster for any questions. Thank you.
Hello, my name is Jenna Epping, and along with my coworker, Dr. Yao, we've been developing a new application for lensless imaging, namely particle image velocimetry. In PIV, we take 2 successive images of particles in a flow field illuminated by a laser sheet or volume. By performing a cross-correlation algorithm on the 2 images, we can compute a quantitative velocity field. In some of our applications, the long working distances and required magnification, along with limited optical access and complex model geometries, can make the setup difficult or impossible. To combat these problems, we started exploring the feasibility of using a lensless imaging setup to image the particle field without a lens. This way, the sensor and mask can be embedded inside of the wind tunnel model to perform near-surface measurements. Using a cheap diffuser-type mask and readily available PIV components, we performed velocity measurements with a benchtop setup, as shown here. Compared with a lensed system, the results agreed well. We were able to recover many of the same particles, and the mean and instantaneous velocity fields were in good agreement.
Hello everyone. You may have the experience of taking panoramic photos with your smartphone. Many of you may have wished that you could take panoramic photos in a single shot without needing to rotate your phone. You might have also wished to take panoramic videos. This poster presentation will show you how to make that possible. Our approach is to double the horizontal field of view of a regular camera by combining the horizontal field of view of 2 of them pointing in 2 directions. We use folded prism optics to achieve parallax-free image stitch. We then seamlessly merged 2 images via high-speed imaging processing on mobile ISP hardware to form a panoramic image. Please come to the post to see the TPC demonstration in live. Thank you.
This is the presentation of our work in microscopy. The microscopy has a trade-off between speed and resolution. It's because the optical throughput is limited. The traditional imaging is either low resolution or low speed, which is not good for biological studies. To solve this problem, we propose a deep learning-based virtual scanning method. It's a robust, generalized network with physical priors. For example, the cells migrated very fast in the mouse liver. We can remove the motion artifacts. The method is also published in Nature Methods. Another case is voltage imaging, which need a very high speed to record. With virtual scanning, we realized ultra-fast 3D imaging at 500 volumes per second. It's a brief summary of our work. Thanks for your listening.
All right, so those are the spotlight posters, which we'll be presenting in just a bit upstairs. And the last two were actually demos, so there's going to be 5 demos, which will take place today and tomorrow. And something to keep in mind while you look at those posters and demos, which are in red, and also the demos, is that there will be an audience vote on Wednesday. So even if you can't get to see them all, you'll have the videos on YouTube. And
Don't leave yet.
you have something to say too?
John has something to say, yes.
Yes, and so yeah, that's essentially it. And we'll give reminders about the audience votes, and we'll see the second half of all the spotlight videos tomorrow. All right, I'll hand it over to John.
Right, turning it over to John. He will say something to the outreach event that is actually happening during the poster session. And let me just actually finish until you can get the coffee afterwards. Don't forget the community drink at 6:30, which is bottom outside on that terrace. So 6:30 is a community drink. The posters will be upstairs. So John, it's all yours.
Thank you, Sabina. I'll keep it short. Okay, I don't want to keep you too long from your coffee. So on behalf of the committee and Emma, myself, the poster chairs, we've organized a few outreach events, a poster and demo session, which is happening immediately after the coffee break. There you may have received emails from some of our rising stars, or some actually already risen stars in the community, asking you to serve as their mentors. For those of you who've responded, actually many of you have responded, thank you. Thank you for giving something back to the community. One of the other things we've done is is to try to kind of expand the outreach, reach out to underserved communities that don't typically attend ICCP. That includes other universities. For instance, we have representation from University of South Florida, myself and my grad student. So, we've also essentially invited students to talk about some of their work in an outreach session around 6 o'clock. So please attend that, give them feedback. These are from people who are not traditionally represented in ICCP. Okay, so hopefully you'll be able to stay back after the poster session and enjoy those posters as well. All right, thank you.